Coming to our next topic, so in carbohydrate metabolism, the most important pathway is glycolysis. So whenever a glucose enters into the cells, so that glucose is trapped as glucose 6-phosphate. Once it is trapped as glucose 6-phosphate, it may enter into glycolysis or it may enter into glycogenesis, so it may be converted into other sugars. So the significance of conversion of glucose to glucose 6-phosphate once it enters into the cell is to trap the glucose in the form of glucose 6-phosphate. Right? And the major way in which glucose is subjected to oxidation is glycolysis. Right? So glucose undergoes oxidation, it will be converted into fats, it will be converted into amino acids, it can be converted into other sugars. So there are different fates for glucose. The main fate of glucose is oxidation of glucose. And the main pathway by which glucose is oxidized is glycolysis. So what is glycolysis? So glycolysis is defined as the breakdown of glucose into pyruvate or lactate in the presence or absence of oxygen is called as glycolysis. So there are a sequence of reactions which break down the glucose into two molecules of pyruvate if oxygen is there, and in the absence of oxygen the same glucose is converted into two molecules of lactate by glycolysis. So we have two types of glycolysis based on the availability of oxygen. If oxygen is there, then that type of glycolysis we call it as aerobic glycolysis, and in the absence of oxygen we call it as anaerobic glycolysis. So these are the two types of glycolysis, aerobic and anaerobic glycolysis. Coming to its site where this glycolysis occurs. This glycolysis occurs in cytosol. Right? So only cytosol has the enzymes concerned with the glycolysis, so that's why glycolysis occurs in the cytosol. Coming to its biomedical importance, there are some tissues which depend upon glucose for its energy. So most important of them are brain and erythrocytes. So erythrocytes, the only source of energy for erythrocytes is glucose, and that too by a process called as glycolysis. So glycolysis supplies energy to erythrocytes. So it cannot derive energy from the oxidative phosphorylation. So the only way in which energy is supplied to erythrocytes is glycolysis. And brain requires glucose. Brain utilizes glucose predominantly as the energy source, and per day brain requires 120 grams of glucose. So that is the significance of glucose as energy source for brain nervous system. And coming to phases of glycolysis. So here we have two phases of glycolysis, phase 1 and phase 2. So phase 1 is the energy investment phase and phase 2 is the energy generation phase. First you are investing the energy, and then energy is derived in the next phase. So basically we have two phases. Phase 1 is the energy investment phase, phase 2 is the energy generation phase. And coming to reactions, so all the reactions of glycolysis, they occur in cytosol, and here the starting material is glucose. So glucose is converted into glucose 6-phosphate by the action of enzyme called as glucokinase or hexokinase. So these two enzymes, they can convert glucose into glucose 6-phosphate. So this step, the first step in glycolysis, requires energy supplied by ATP. So ATP transfers the phosphate group to the glucose, and that phosphate group is attached to the 6th carbon of glucose. So that glucose is converted to glucose 6-phosphate. So this is a step where ATP is required. So we are investing energy in the initial stages of glycolysis, and the first step is conversion of glucose to glucose 6-phosphate. Now glucose 6-phosphate is isomerized to fructose 6-phosphate by the action of enzyme called as isomerase, phosphohexose isomerase. So that isomerase converts glucose 6-phosphate to fructose 6-phosphate. Now fructose 6-phosphate in the next reaction catalyzed by another kinase called as phosphofructokinase converts fructose 6-phosphate into fructose 1,6-bisphosphate. So this is the second step in glycolysis where ATP is required. The first step and the third step, they require the ATP. So that's why we call this phase as the energy investment phase. So by the action of enzyme phosphofructokinase, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. So one more phosphate is taken from the ATP and it is now added to the first carbon of fructose, so that it becomes fructose 1,6-bisphosphate. Two phosphates at the first carbon and the sixth carbon. Right? Now, what is the fate of fructose 1,6-bisphosphate? This fructose 1,6-bisphosphate undergoes lysis by the action of enzyme called as aldolase. So aldolase breakdowns this fructose 1,6-bisphosphate into one molecule of glycerol 3-phosphate and one molecule of dihydroxyacetone phosphate. So 6 carbon fructose 1,6-bisphosphate is now converted into two 3-carbon compounds, and glycerol 3-phosphate, dihydroxyacetone phosphate, these two can be interconverted by the action of enzyme called as isomerase. Triose phosphate isomerase converts dihydroxyacetone phosphate into glycerol 3-phosphate. So now we have two molecules of glycerol 3-phosphate. These two molecules of glycerol 3-phosphate in the next step by the action of enzyme called as glycerol 3-phosphate dehydrogenase is converted to 1,3-bisphosphoglycerate. Right? So this is a step in glycolysis where NADH is generated, the conversion of glycerol 3-phosphate into 1,3-bisphosphoglycerate. So in this reaction, one phosphate is added to molecule, so that is converted to 1,3-bisphosphoglycerate. So this has two phosphates as the first position and third position, and this 1,3-bisphosphoglycerate is a high energy compound. This in the next reaction undergoes substrate level phosphorylation and is converted to 3-phosphoglycerate by the action of enzyme called as bisphosphoglycerate kinase. So 1,3-bisphosphoglycerate kinase converts 1,3-bisphosphoglycerate into 3-phosphoglycerate. Here one ATP is generated, and this type of ATP generation directly from the substrate is called as substrate level phosphorylation. So ATP is produced in two ways. The major way is oxidative phosphorylation and the other way is substrate level phosphorylation. So when ATP is formed directly from the substrate without the involvement of electron transport chain, we call such type of phosphorylation as substrate level phosphorylation. So this is the first step in the glycolysis where ATP is synthesized from the substrate level phosphorylation. Now this 3-phosphoglycerate is now converted to 2-phosphoglycerate by the action of enzyme called as mutase. So there is shift of phosphate from the third carbon to second carbon, so that 3-phosphoglycerate is converted to phosphoglycerate. 2-phosphoglycerate in the next reaction catalyzed by the enzyme enolase is now converted to phosphoenol pyruvate. So that phosphoenol pyruvate in the next reaction catalyzed by the enzyme called as pyruvate kinase is converted into pyruvate. So this is how pyruvate is formed from glucose in aerobic glycolysis. So the end product of glycolysis, the end product of aerobic glycolysis is pyruvate. And if it is in the absence of oxygen, that pyruvate is now further converted into lactate by the action of enzyme called as lactate dehydrogenase. So that lactate dehydrogenase converts pyruvate into lactate, lactate into pyruvate. So interconversion is brought by lactate dehydrogenase. As usually the conversion of pyruvate to lactate occurs in the absence of oxygen. So the conversion of pyruvate to lactate, this type of glycolysis we call it as anaerobic glycolysis. If it stops at pyruvate, we call it as aerobic glycolysis. So in the conversion of phosphoenol pyruvate to pyruvate, one more ATP is produced directly from the substrate. So this is the second step where ATP is produced directly from the substrate, and this is second step of in glycolysis where substrate level phosphorylation occurs. So this is about reactions of glycolysis. So we have started with a 6-carbon molecule glucose, and the end product is 2 molecules of pyruvate in the presence of oxygen, two molecules of lactate in the absence of oxygen. So these are the reactions of glycolysis. And coming to regulation, how this glycolysis is regulated. This glycolysis is regulated by three mechanisms, that is induction, repression, and the second one is allosteric regulation, and the third one is covalent modification. So these three mechanisms, they regulate the glycolysis, rate of glycolysis. Coming to first one, induction and repression. Whenever the concentration of glucose is more in the blood, then insulin is secreted from the pancreas. So that insulin activates all the key enzymes of glycolysis. So it increases the synthesis of key enzymes of glycolysis by increasing the activity of genes which code for corresponding proteins. So this type of regulation which occurs at the gene level is called as induction and repression, right? So insulin increases all the enzymes which are concerned with the increases the concentration of all the key enzymes of glycolysis. So key enzymes of glycolysis are hexokinase, phosphofructokinase, and pyruvate kinase. So these three enzymes are called as key enzymes of glycolysis. And here the effect of insulin is when insulin increases the activity of genes which code for these three enzymes, the concentration of these enzymes, key enzymes, is increased. So whenever the concentration of key enzymes is increased, the rate of glycolysis increases. So this is about the induction and by which the rate of glycolysis is increased. Now coming to the second process, allosteric regulation. So here the key enzymes, the activity of key enzymes of glycolysis is increased whenever the concentration of AMP is more. So AMP activates the key enzymes of glycolysis, that is called as allosteric regulation. So most important allosterically regulated enzyme in case of glycolysis is phosphofructokinase. The activity of phosphofructokinase is increased whenever there is increased concentration of AMP. So AMP increases the activity of phosphofructokinase, and this is a positive regulator, positive modifier. The same way ATP, whenever the concentration of ATP is more, then ATP inhibits the phosphofructokinase activity. So this is allosteric regulation. Coming to covalent modification. So the key enzyme, one of the key enzymes, pyruvate kinase, it is inactivated whenever phosphate group is added to that pyruvate kinase. So phosphorylation of pyruvate kinase decreases the activity of pyruvate kinase. So overall rate of glycolysis decreases. So whenever the phosphate group is removed, that you call it as dephosphorylation, the activity of pyruvate kinase increases. So this is covalent modification. Right? So by these three mechanisms, rate of glycolysis is regulated. And coming to inhibitors, there are some inhibitors which inhibit the enzymes of glycolysis. For example, when you take enolase is inhibited by fluoride. So that is one inhibitor. Fluoride inhibits the enolase, and the another inhibitor which inhibits the enzyme of glycolysis is iodoacetate, inhibits glyceroldehyde 3-phosphate dehydrogenase. So these two are the inhibitors of glycolysis. And coming to energetics. So the main purpose of oxidation of glucose is to generate energy. So how much energy is generated when a single glucose molecule is oxidized by glycolysis? So that we'll see here. So here you can see that in the first reaction and the third reaction, ATP is utilized, and that phase we call it as phase 1 reactions, where energy is supplied. But after that, conversion of fructose 1,6-bisphosphate into glyceroldehyde 3-phosphate and dihydroxyacetone phosphate. From there onwards, the molecule is prepared for release of energy. So here, the first substrate level phosphorylation, that is conversion of glyceroldehyde 3-phosphate into conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate, generates 1 ATP. So that step we call it as substrate level phosphorylation. And the another reaction where the ATP generated is conversion of phosphoenol pyruvate to pyruvate. So two ATPs are formed in the glycolysis in the energy generation phase, and when you consider two molecules of glyceroldehyde 3-phosphate, now it is equal to 4 ATPs, and so total 4 ATPs are produced in the phase 2, that is energy generation phase, and 2 ATPs are utilized in the phase 1 energy investment phase. So the net gain of ATPs in glycolysis is 2 ATPs. Right. So when you consider NAD, which is produced in the conversion of glyceroldehyde 3-phosphate into 1,3-bisphosphoglycerate, so that NADH when enters into the electron transport chain, it produces the ATPs equal to 2.5 ATPs, and according to old calculations it is 3 ATPs. So when you consider that, so we'll get 3 ATPs from each NADH, and when 2 NADH enter into electron transport chain, it generates 6 ATPs. So 6 ATPs from NADH and 4 ATPs from the substrate level phosphorylation, total we will get 10 ATPs, and whereas 2 ATPs are utilized in the energy investment phase, so minus 2, it gives rise to total 8 ATPs. So net gain of ATPs in aerobic glycolysis, when you consider NADH, it is 8 ATPs. The only ATPs is, so the net gain of ATPs is 2 ATPs. So you can consider, when you consider NADH, it should be 8 ATPs, and without NADH, you will get 2 ATPs. So that's about the energetics of glycolysis. Now coming to another pathway, gluconeogenesis. So glycolysis is breakdown of glucose into pyruvate. Here the reverse occurs, gluconeogenesis. Gluco means glucose, neogenesis, synthesis. So you are synthesizing glucose from new compounds. So that's why it is called as gluconeogenesis. So formation of glucose from non-carbohydrate sources is called as gluconeogenesis. Right. So this occurs partly in the cytosol, partly in the mitochondria. It occurs in the two compartments, cytosol and mitochondria. And when you see the reactions, here we'll start with the pyruvate, because so pyruvate acts as the precursor for the synthesis of glucose. So pyruvate, which is formed in the glycolysis, enters into the mitochondria, and that pyruvate here is converted into oxaloacetate by the action of enzyme called as pyruvate carboxylase. So this reaction requires ATP, and the coenzyme required in this reaction is biotin. So carboxylation reaction, coenzyme required is biotin. So that pyruvate by action of enzyme called as pyruvate carboxylase is now converted into oxaloacetate in the mitochondria. So that mitochondria can be converted into phosphoenol pyruvate, but so phosphoenol pyruvate cannot cross the mitochondrial membrane, so that we have to convert the phosphoenol pyruvate, so we have to convert that oxaloacetate into phosphoenol pyruvate. So in order to shift the oxaloacetate from the mitochondria into the cytosol, so we require the help of malate. So that oxaloacetate actually, which cannot pass through that mitochondrial membrane, it has to be converted into malate. So that is brought by an enzyme called as malate dehydrogenase. So this occurs in the mitochondria. We call it as mitochondrial malate dehydrogenase, converts oxaloacetate into malate. So that malate now can pass through that mitochondrial membrane into the cytosol. So malate now, which entered into the cytosol, can be reconverted into oxaloacetate by the action of enzyme called as cytosolic malate dehydrogenase. Right? Now oxaloacetate is formed. So that oxaloacetate can be converted into phosphoenol pyruvate, and that is brought by an enzyme called as phosphoenol pyruvate carboxykinase. So this step requires GTP, and there is removal of carbon dioxide in this reaction, so that oxaloacetate is now converted into phosphoenol pyruvate. Now what happens to that phosphoenol pyruvate? That phosphoenol pyruvate is now converted into 2-phosphoglycerate by the action of enzyme enolase. 2-phosphoglycerate is converted into 3-phosphoglycerate by the action of enzyme mutase. 3-phosphoglycerate is converted to 1,3-bisphosphoglycerate by the action of enzyme called as 1,3-bisphosphoglycerate kinase. So that 1,3-bisphosphoglycerate in the next reaction is converted into glyceroldehyde 3-phosphate by the action of enzyme called as glyceroldehyde 3-phosphate dehydrogenase. Now, two molecules of glyceroldehyde 3-phosphate they form. So they are formed. So one of them is converted into dihydroxyacetone phosphate. So glyceroldehyde 3-phosphate, dihydroxyacetone phosphate, they react to form fructose 1,6 bisphosphate. So that fructose 1,6 bisphosphate, which is formed from reaction of glyceroldehyde 3-phosphate and dihydroxyacetone phosphate by the action of enzyme aldolase, is now converted into fructose 6-phosphate. So for the conversion of fructose 1,6 bisphosphate into fructose 6-phosphate, we require an enzyme called as fructose 1,6 bisphosphatase. So that removes the phosphate from the fructose 1,6 bisphosphate so that it is converted to fructose 6-phosphate. So this is the enzyme different from that of glycolysis. So actually in glycolysis we have enzyme, separate enzyme which converts fructose 6-phosphate into fructose 1,6 bisphosphate, that is phosphofructokinase. Here this is a different enzyme, that is fructose 1,6 bisphosphatase. So once it is converted into fructose 6-phosphate, it is isomerized to glucose 6-phosphate by the action of enzyme called as isomerase. Now glucose 6-phosphate by the action of enzyme glucose 6-phosphatase is converted into glucose. So this is how pyruvate is converted into glucose. So non-carbohydrate sources, they can be converted into glucose. So this whole mechanism we call it as gluconeogenesis. Right? So these are the reactions of gluconeogenesis. Now coming to substrates for gluconeogenesis. So as we discussed, we are synthesizing glucose from non-carbohydrate sources. So what are those non-carbohydrate sources? Those are pyruvate, propionyl-CoA, lactate, alanine. So these are the non-carbohydrate sources from which glucose can be synthesized. So pyruvate, so just now we have seen how pyruvate is converted into glucose. And coming to propionyl-CoA, so that propionyl-CoA which is formed from the oxidation of odd chain fatty acids is utilized in the synthesis of glucose. So propanyl-CoA is converted to D-methylmalonyl-CoA and D-methylmalonyl-CoA is converted into L-methylmalonyl-CoA. So that L-methylmalonyl-CoA can be converted into succinyl-CoA, and that succinyl-CoA now enters into the TCA cycle where it is converted into succinate, succinate to fumarate, fumarate is converted into oxaloacetate, and that oxaloacetate now from mitochondria is transported into the cytosol as malate, and that can be converted again into the phosphoenol pyruvate. And this is how propionyl-CoA, which is formed from the oxidation of odd chain fatty acids, can be used as the gluconeogenic source. Coming to lactate and alanine. So lactate, which is formed in the muscles and erythrocytes, it can be converted into the glucose. So there is one reaction that converts lactate into pyruvate, that is lactate dehydrogenase. So that lactate dehydrogenase converts lactate into pyruvate. So once it is converted into pyruvate, the same set of reactions will occur in the conversion of pyruvate into glucose. Okay. And coming to alanine, so alanine is most of the alanine is found in the muscle. So that alanine which is formed in the muscle by a process called as transamination. So alpha ketoglutaric acid takes up the ammonia. So in this reaction, by the action of enzyme called as alanine transaminase, it forms a compound called as a type of amino acid called as alanine. So that alanine can be used in the synthesis of glucose. Alanine formed in the muscle is transported into the liver through blood. So once it reaches the liver, that alanine by the action of enzyme alanine transaminase is converted into pyruvate. So that pyruvate again by a sequence of reactions can be converted into glucose. So this is how lactate and alanine can be converted into glucose. So lactate, which is formed in the muscle, enters into the liver, and in liver it is converted into glucose, and that glucose again reaches to muscle, is utilized in the glycolysis. So this cycle is called as Cori cycle. And the same way, alanine, which is formed in the muscle, can be converted to glucose in the liver, and that is again can be supplied to the muscle. So this cycle is called as glucose alanine cycle, conversion of lactate into glucose conversion of alanine into glucose. So these two cycles are Cori cycle and glucose-alanine cycle. And this, coming to regulation of gluconeogenesis. So gluconeogenesis can be regulated again by the same process as you have seen in case of glycolysis: induction depression, covalent modification, and allosteric regulation. So induction depression involves regulation brought by glucagon. So glucagon increases the activities of all the key enzymes of gluconeogenesis by increasing their synthesis. So it increases synthesis of key enzymes of gluconeogenesis. So here glucagon increases synthesis of key enzymes of gluconeogenesis and decreases the expression of key enzymes of glycolysis. So it induces the enzymes of gluconeogenesis, it represses the enzymes of gluconeogenesis. So that we call it as induction and repression. Coming to covalent modification, here in case of gluconeogenesis, so there is one enzyme called as phosphofructokinase 2 and fructose 2,6-bisphosphatase. So these two enzymes are present on the same polypeptide chain. So we call it as tandem enzyme. So whenever the tandem enzyme is phosphorylated, means whenever you phosphorylate fructose 1,6-bisphosphatase, so phosphorylation of that enzyme increases the activity of fructose 1,6-bisphosphatase, and whenever the fructose 1,6-bisphosphatase activities increase, then the concentration of fructose 2,6-bisphosphate decreases. So fructose 2,6-bisphosphate actually increases the activity of phosphofructokinase. So whenever the concentration of phosphofructokinase, whenever the concentration of fructose 2,6-bisphosphate is low, the activity of phosphofructokinase is decreased. So whenever the phosphofructokinase activity is decreased, the other enzyme, the rate of glycolysis decreases and the rate of gluconeogenesis increases, because the activity of the enzyme which is catalyzed in the same reaction in gluconeogenesis is increased. So this is how covalent modification increases the rate of gluconeogenesis. Coming to allosteric regulation, in case of allosteric regulation, when you see pyruvate carboxylase, the enzyme which converts pyruvate into oxaloacetate, is allosterically activated by acetyl-CoA. So whenever the concentration of acetyl-CoA is more, that acetyl-CoA activates the pyruvate carboxylase, one of the key enzymes of gluconeogenesis. So whenever the activity of that particular enzyme is increased, then the rate of gluconeogenesis increases. So these are the three ways in which gluconeogenesis is regulated. So